combine the inherent link between temperature and water vapor. Correct?
Yes.
Forgot where I was going with this.
So much for not having a lot of editing to do.
Well, hello and welcome into episode 6 of LI-COR's podcast series Leaf to Leaf. I am still your host, Seton Bockel. They let me keep doing this. We are here to discuss 2 different chapters today, 7 and 8 of The Hitchhiker's Guide to Leaf Level Gas Exchange. And we are going to be talking about water vapor and temperature. But before we dive into that hot mess, that was a joke. I'm here with a guest who needs to introduce himself.
I'm Jason Hupp, Director of Science Research and Applications here at LI-COR and one of the authors of A Hitchhiker's Guide to Leaf Level Gas Exchange.
How are things?
Oh, things are lovely. The weather's gotten a little colder than I like. It has made the cycling into the office a little less than fun the past couple of days.
If it drops below 60, I think you turn blue.
I do, yeah. It's very unpleasant. I'm not well adapted to the Nebraska climate.
Florida man at heart?
All the way into my soul.
Well, it's funny. For those of you who may not know, LI-COR is located in Lincoln, Nebraska, which is like renowned for a few things, football, and then really like having the worst representative weather every season.
I also like the phrase about Lincoln that it is equidistant to everything, but close to nothing.
That's a great t-shirt.
Isn't it?
That's a pretty good t-shirt. But honestly, fall is like, I don't know, a week long, and they're not back-to-back days. It's like throughout the year, you get about 10 days of fall.
Yeah, that sounds right.
Yeah, and I think we've already gotten like 4 or 5 of them. But summer is super uncomfortable, right? It's hot and it's humid. And the joke in July and August here is, well, at least it's a wet heat. And as I was out in a cornfield 2 days ago, I was reminded with how uncomfortable it is, and it was kind of befitting that we're going to be talking about water vapor and temperature this episode. And so we're going to be talking about both of them, but we're going to maybe just break it down one and then the other, as much as these are inherently linked. Let's just start with water vapor. Why is talking about water vapor so difficult to do by itself or difficult to characterize?
Because it's confounded by temperature. It almost seems silly to split them into 2 chapters.
It does. Blame the author.
Yeah. Man, whatever possessed him to do that is unfathomable to me.
That guy's the worst.
Yeah. Yeah, it's confounded by temperature, and there's a whole bunch of different ways to express it. CO2 is easy, right? Like we just use moles per moles, and there's other ways to express it, but they're all related to effectively moles per mole. Water vapor, we can use mole fractions, we can use molar densities, but then there's all these other weird metrics that show up. Like everybody loves relative humidity. We use relative humidity a lot in chapter 3 because people feel very comfortable with it, even though it's absolutely the wrong metric to use in our application.
Why do we have to hate on RH?
Well,
for those who are listening, which is everyone, I guess.
Let's see.
It's present. I got the dirtiest look that I've received in a long time.
I wasn't part of Seton's interview process, so we'll see how he does with this.
No, this isn't about me. That's actually I was getting to. That was one of my interview questions.
It was one of my interview questions too.
Yeah.
It's a big deal here, right? Like we always ask people, like, can you explain relative humidity? And
my answer was, that's a deep question, and it's all relative. So is there really one answer?
Wow, and we still hired you for that.
It was an employee's paradise.
I see.
Yeah.
Well, I can tell you lots of people get it wrong because they miss that it's, relative to how much the air can hold at a given temperature. And as air temperature changes, obviously, it can hold different amounts of water vapor. And so you get these changes in RH that don't necessarily reflect any change in the amount of water vapor that's actually in the air.
But so, okay, why do we use it so much? What is the draw to it?
I don't know where that starts from. I don't know the history of relative humidity. Maybe that's a whole separate podcast topic.
Sweaty thoughts.
Ooh, it could be a whole series.
All right, calm down, audience. This isn't definite yet. But no, it seems interesting when there are other methods that probably, wink wink, are a lot more appropriate.
Yeah, sure. So we lay out 5 or 6, I have to think about that, metrics for water vapor and air in the book. So we talk about mole fraction, we talk about relative humidity, we introduce specific humidity, absolute humidity, vapor pressure deficit, and we kind of give a physical basis for all of these. And then, we circle all the way back to chapter 3 and kind of plug them into a very basic fixed law formulation for transpiration.
Right.
And then we look at what happens if you hold any one of these metrics constant during your measurements from a theoretical perspective, right? Assuming no biological change. And show how transpiration changes as a function of them. And spoiler, for one of them, transpiration is a flat line. And for all of the rest, it's like a crazy curvy line, right? It's like there's a super strong dependence. And this gets us to like this discussion that justifies this thing that we call vapor pressure deficit, or leaf vapor to air vapor pressure deficit, as our like greatest metric to describe water vapor for gas exchange measurements.
Right. And that's one that we definitely promote here. And I don't want to, people should go read the chapter, but maybe give a synopsis, give us like the bullet points. Why VPD? Why is that
the
best?
It's an expression of water vapor in the leaf relative to water vapor in the air. So when you're talking about a process that is driven by a diffusion gradient between the leaf and the air, that metric encompasses both sides of the diffusion gradient. And so when it's the thing driving the process that you're interested in is the diffusion gradient, if you're holding that metric that describes the diffusion gradient constant, you're holding the diffusion gradient constant.
That was very well put. That was very well put. And
he seems very surprised by this, actually.
I may have stopped recording to tell him that was nicely done. But to be fair, I wanted to get it on recording so he could use this against me later. If we're thinking about this diffusion gradient, what controls that gradient, that diffusion gradient? So I guess I'll ask this question: if stomata are the gatekeepers, then who are the key masters?
Ooh,
and if you don't know what we're talking about or why that joke was funny, because it was, then you should Google it.
Yeah, I mean, jokes are always funnier when you have to explain them, but
that's how I live my life. You saying that's not funny?
That suddenly explains a lot.
Oh, I've got a lot of reflection to do.
Well, I think the key master here, on the leaf side, I mean, you could argue at some point it becomes a leaf water status question. But really, for most of the things that we're interested in, it's temperature. And it's leaf temperature that sets the amount of water vapor inside the leaf. And it's temperature of the air and the availability of water that set the amount of water vapor in the air outside the leaf.
We'll get back to the podcast after a word from our sponsors. This isn't just growing. This is training. Every stomata, every leaf, every cell pushed to the limit with VPD Elite. You think you know consistency? Try locking in the perfect vapor pressure deficit. Not once, not twice, but every single day. This is precision that sweats with you, grows with you, and wins with you. VPD Elite. This is more than science. This is you versus you, and your plants are ready. So those are both super important, but even between leaf and that ambient gradient, I'm trying to get you.
Are you getting to the boundary layer?
Why, yes, I am.
A layer that represents the boundary between the two.
What role from an instrument? Let's break this down. From an instrument standpoint, what's the importance here?
The importance is that we manipulate a boundary layer in the instrument. I think that's the direction you're getting at here. This is an important topic in the gas exchange measurement versus just the physical process of stuff. If you think back to chapter 3, we talked about like how do we get to stomatal conductance from a measurement of transpiration. We introduced this parameter, the boundary layer, and we said we have to know something about it to get to stomatal conductance. Here, we talk about in depth, like how do we know something about the boundary layer and how is it different inside the chamber versus outside of the chamber? I think one of the neat analyses that we do in there.
Preface.
Oh man,
Bring
this
it back.
tomato,
Tomato, pineapple.
yeah, that's all equivalent. Is we show what happens to transpiration as we would measure it in a chamber at different boundary layer conductances, sort of like a leaf in the same physiological state inside the chamber versus a leaf in the same physiological state outside the chamber under sort of typical atmospheric conditions.
Okay, so from that instrument standpoint, how does that lead into the biological reality?
Well, the reality of it is that, as we've established early on, like the parameter we care about is stomatal conductance. That's why we call it apparent transpiration, not real transpiration. And it means that in a measurement, like we're always trying to reproduce boundary layer conditions for water vapor inside the chamber versus bulk atmosphere conditions.
Right.
And the boundary layer is always wetter than the bulk atmosphere.
So keeping in mind the biological ramifications and what the instrument can do, what sort of manipulations, like if someone was listening to this and read the chapter, what are some things that they could do to realize this in somewhat real time? We're talking about water vapor and temperature. Work with me. Help me help you. If you don't know that movie, also Google it.
Meaning, how should you set up a chamber?
How would, yes. If
okay.
one would, yeah. That wasn't clear.
It was very unclear. I was trying to
me.
get at what Seaton was asking here, and it's hard to tell with him sometimes.
I was dropped a lot as a child.
I mean, me too. That's what makes me so slow. It's a product of the 1980s. Yeah, I mean, the idea is that we want to make the chamber wetter than whatever the bulk atmosphere is, right? So that means that we're either humidifying the airstream before it goes in, we're slowing down the air as it goes through the chamber to get the leaf a chance to humidify the air for itself. But we're always working at your favorite metric of humidity that's above ambient. My personal preference is VPDL, and I'm shooting for a value between 1 and 1.5 kPa.
So I'm the reason I'm kind of drilling into this is periodically we have requests and users want information on how to create custom chambers. And with the idea of mixing volume, maybe talk a little bit about that.
Yeah, I mean, the boundary layer in the chamber is driven by turbulence inside the chamber, which is very much a function of how air moves through the chamber. In sort of our traditional chambers, right, there's a mixing fan that's creating a lot of mixing inside the chamber and very high turbulence inside the chamber. I think we give an example in the chapter on CO2 where we show like a time constant in the chamber for the mixing fan that's like, a few tenths of a second. Like it's super fast.
Yeah.
And the consequence of that is that we make the boundary layer around the leaf very thin, boundary layer conductance very big. Doesn't matter so much in our stomatal conductance calculation.
Sure.
We still know what it is. We still factor it out.
Is it relative?
It's relative to something. The wind speed.
See, this is exactly how I answered the question during my interview. I had that same look on that you have on your face right now as well. But if we're getting to this efficiencies, which are somewhat relative, right?
Yeah.
We have it's kind of a hot topic. No, not the store and the mall, but the hot topic of water use efficiency.
Oof. Yeah.
Why oof?
That's my least favorite 3 words when it comes to gas exchange measurements. Man, people see assimilation and transpiration and they just want to divide one by the other and call it instantaneous water use efficiency like it's the greatest thing since sliced bread. And we do a nice job of demonstrating why that's garbage in a leaf level gas exchange measurement in the book. Pretty proud of that. there is a better metric, right? Like we've
Yeah.
talked several times that, stomatal conductance is a metric of water flux, but it's like transpiration standardized by the diffusion gradient. And you can take assimilation and divide it by stomatal conductance and calculate something that we call intrinsic water use efficiency, which removes these confounding effects from leaf temperature and chamber humidity from your water use efficiency metric. There's a really nice figure that shows like, here's 3 data sets collected by 3 different people on 3 different species. It's our favorite corn data set in there. And then there's some wine grapes that I collected out in the Willamette Valley.
Ooh.
Yeah, it was very exciting. And then some data from a pak choi that I think Doug collected.
That sounds like something Doug would do.
Yeah, Doug was on a few podcasts ago. He's great at collecting data, amongst other things. Anyway,
growing bok choy.
3 different people, 3 different crops, 3 different data sets, 2 different instruments. Some of it's with a 6800, some of it's with 6400. And all of them were done using sort of typical best practices, but they were all done using different ways of controlling water vapor in the chamber. And the figure's great. There's one panel that shows like instantaneous water use efficiency that doesn't make any sense. It's like a scatter plot of scatter. And then all we do on the next plot is show it as intrinsic water use efficiency, and all of a sudden everything collapses to like perfectly straight lines.
So this begs the question, why? Why are people collecting instantaneous?
It's a question of understanding sort of the biophysics of what you're measuring versus just the biology of what you're measuring.
Okay.
And I think if you have a biophysical perspective on it, you look at it and you go, man, instantaneous water use efficiency doesn't make any sense in this case. If you're just looking at it from the biology, it's like, oh man, this makes perfect sense. But, a leaf chamber is not the bulk atmosphere. And we have to recognize that there are some things different there.
I think you broke that down really well. And something that was said several times during your explanations was referring back to chapter 3.
Yeah.
And so if you haven't read chapter 3, please go back and read it. I think it'd be incredibly helpful. In chapter 3, we emphasize the energy balance. You have some great mathematics in chapter 3, as well as the start to, I believe it's chapter 8.
Yeah. I mean, I do love the maths.
Yeah. Radiative heat flux.
Oh, yeah. Well, chapter 3 gives sort of like a hand-wavy introduction to energy balance.
Mm-hmm.
And in chapter 8, we set down and derive it from first principles.
Deep
it's
dive.
So exciting.
There's a lot of math.
Yeah. And assumptions. We love assumptions. Math and assumptions. The two go hand in hand, actually.
So, yeah, I mean, you break down radiative loading, irradiance incident on the leaf, absorption. Some of these we've covered in other chapters. But
we kind of bring it all in together to show how, like, light, not just light that we see, but light energy in general, like, affects leaf temperature.
Right.
And then we sort of derive the sensible heat flux. Not sort of, we do. We show how turbulence around the leaf is a pathway for energy exchange. And then we show the conversion of transpiration to the latent heat flux. And then we kind of bring all these things together to say, like, not only do all these things affect leaf temperature, but here's how we estimate leaf temperature if we know these other parameters, processes.
Yeah.
Yeah.
So there was a lot of language that you used, a lot of vocab words that would be found in the glossary of most books.
We don't have a glossary. No.
No.
We do have like a 10-page long appendix of symbols.
I was just going to read those aloud. That was going to be the last episode of this podcast, was me describing what the symbol was and then reading the definition.
I don't even know how you pronounce all of them, so I will be tuning in to listen to that as well.
It'll be, sounds like triangle. I don't know. So latent heat, sensible heat, like these are things that I think we kind of toss around internally. Like we all know what we're talking about. But for users, for listeners that may be less familiar, can you break down some of those broad differences between these? Because, like, is sensible heat flux the same thing as latent, but with better judgment and more practicality?
Well, I think they call it sensible because it's a flux that you sense,
Oh, I see. Very
like
fine.
as in a sense of warm and cold. I mean, it's just a physical exchange of energy driven by like turbulence. Latent heat flux is, I mean, it's evaporation, right? Like,
Yeah.
you've probably heard the term latent energy of evaporation or latent heat of evaporation. That's our latent heat flux. So instead of expressing transpiration like in moles per meter squared per second, as we have everywhere else, we're going to express it in watts.
And that is a sight to behold. Do we break down that math for them?
Oh, we do.
Oof.
Yeah.
Some good. That is some good reading.
It's just a constant that you multiply it by.
Don't worry. No, don't give it all. Well, this episode's over. Just ruined it.
Which, much like my phone number, I can't remember without having it written down in front of me. So
I've seen it. It's weird. You
Yeah.
can't do it. We'll get back to the podcast after a word from our sponsors. You're a scientist. You can recall every step of the Calvin Benson cycle, but still can't remember your own phone number. But forgot trash day again? That's why there's Lab Brain Daily, the supplement designed not for your scientific brilliance, but for the small, ordinary things that matter outside the lab. Side effects may include remembering embarrassing grad school emails, old comments from a reviewer two, and every bad joke your PI ever told. Not approved by the FDA, NSF, or your department chair. Lab Brain Daily, because even nerds deserve groceries too. So moving away from just the pure mathematics and focusing though on temperature as a whole, gas exchange systems are all about control.
Yeah.
So what's going on in the chamber, trying to match it, trying to manipulate it.
I think this is one of the cooler discussions of an environmental control, like how we actually do it in an instrument. Because I think it's the one that probably most people are least familiar with. Like, how do you actually regulate temperature?
Right. Well, there's different locations, at least in the
There's huge
6800.
temperature gradients throughout the system. There's
Right.
different places you could control. There's different things that you target. And then, how do you do that control? Like, we're not walking around with like a portable air conditioner unit. So how do we cool the chamber down?
Sure.
I think the way that we control temperature is super neat. I remember when I first learned about the Peltier effect, like I was like, oh, this is the coolest thing I've ever heard of. We talk in, I guess probably back in chapter 3 about how a thermocouple works, and we introduced the Seebeck effect, right? Like you put two dissimilar metals in a loop, and if you put a temperature gradient across them, you get a flow of current. The Peltier effect is just that in reverse. You put two dissimilar metals in a loop, and you flow a current through it, and you get a temperature gradient. And so we have these little solid-state cooler heater devices in there that are called Peltiers or thermoelectric coolers that we can use to drive temperature up and down, and that's all neat. But then there's what we're actually controlling the temperature of and how well that's coupled to the thing that we're trying to control.
Right. And so there is a diagram in the chapter. I think it's a cross-sectional diagram of the erga,
Yeah.
or at least components of it. It shows a lot, if not all, of the locations in which we are measuring and monitoring temperatures.
For sure.
There's also the aspect of what can be controlled, right? We're monitoring temperatures, and that's from a certain standpoint, that's a great diagnostic tool, right? But from a control aspect, from a user's point of view, where are the optimal locations to control set temperature?
Well, I mean, the optimal location would be the leaf temperature, but nobody really does that because how do you control the temperature of the leaf?
You talk to it very sternly.
Yeah,
This
that
is not
doesn't work.
Yeah, it doesn't?
Yeah, no, I've tried. In the case of like the 6800, the temperature control mechanism is built into the back of the mixing fan. And so like when we want to control leaf temperature, we control the temperature of the block the mixing fan's in.
Mm-hmm.
And that leads to some sensible coupling to the air in the chamber. And then there's some sensible coupling between the air in the chamber to the leaf. And so it's a multi-step process, which is why temperature control of the leaf is never fast.
Sure. And expensive from an energy standpoint, right? I think if anyone has tried to control leaf temperature, it sounds like your instrument's going to launch itself to the moon.
Yeah, in that energy balance discussion at the start of the chapter, we go through the relative magnitudes of all of these different components of the energy balance, right?
Right.
And sensible is by far the tiniest of all of them. Yet that's the one that we have to rely on 2 exchanges of to be able to control leaf temperature.
I think most people do advocate controlling for leaf temperature, but we allow a few different options.
Sure.
Why?
Well, different options make sense in different contexts.
Okay.
Like if I'm going to go make a light response curve, what happens in a light response curve? Well, I change my light intensity, my radiative heat flux at every point in that curve. That's a time where I always control on leaf temperature directly, right? Like I want to compensate for the changing radiative load as I go through the curve, so temperature is constant. If I'm going to go do a CO2 response curve, my radiative flux is going to be constant. Depending on how I do the curve, there's probably going to be some change in stomatal conductance, so there's probably a little bit of change in the latent heat flux, but it's probably not huge unless I did the curve really poorly.
That never happens.
Not to me.
Okay, well, that just seems like you're pointing the finger.
So that's a case where I might sort of set the instrument up to control the exchange temperature, the exchanger, that block that the mixing fan's in, let everything stabilize under my normal conditions, and then just let it stay that way, or switch it to keep the leaf at that temperature as I go through the curve. I might not drive it to a specific value.
Right.
We do talk specifically about temperature in the context of ACI curves in there, because all those models that we introduced in Chapter 6 sort of implicitly assume leaf temperature is 25 degrees C.
It's not?
Depends on how you made the measurement.
Yeah.
So we talk about how you adjust measured parameters to sort of theoretical model parameters using these like activation, deactivation energies and another model, basically.
Another model.
Mm.
So the chapter does break down some general impacts. The temperature on gas exchange parameters, right? Carbon. There's some examples for carbon assimilation and stomatal conductance and things. There are also figures that combine the inherent link between temperature and water vapor. Correct?
Yes.
Forgot where I was going with this.
So much for not having a lot of editing to do.
I was doing really well until it epically failed.
Yeah, in both chapters, we go through sort of biological response to temperature, biological response to water vapor. The discussions here are more nuanced than maybe in the earlier chapters where there was like a direct link between the physiology that we're measuring and the parameter that we're manipulating. Whereas there's a lot of physicality in the relationships here, right? Like
that seems borderline inappropriate.
Not in the way that I meant it. Yeah.
Okay, carry
on. Like for water vapor, right? Like there's no direct sensing of water vapor by carbon assimilation or something that drives it, right? So there's like this diffusive linkage. And then the whole like water vapor transpiration is like very physically confounded, less biologically. And then water vapor and stomatal conductance, right? There's both a physical and a biological. People talk about the wrong way response, right? Like where you get this artifactual movement in the guard cells when you change VPD. And temperature is sort of the same thing. There's nobody's shown a definitive biological sensing mechanism for like the stomata to temperature, but you see stomatal response to temperature, but it's probably all like driven by this physical confounding of the diffusion gradient by temperature as opposed to some like actual biological response.
Right. And so it is a non-uniform response, and Doug and I had talked about uniformity in the context of light a couple episodes ago, but in the context of temperature and water vapor, there's some pretty cool examples in this chapter about the non-uniformity of how those are impacting a leaf.
Oh yeah, we do show like beyond the control of temperature, right? Like temperature is an important measurement. Leaf temperature, we have to measure that to get to stomatal conductance.
Yep.
Again, as described in chapter 3. And in chapter 5, as Seaton points out, we introduce these scenarios where we like artifactually make a light non-uniformity of some type in the chamber, and we look at like, oh, well, how would that affect assimilation given a certain response to light? In this chapter, we revisit those scenarios and we say, well, what is that non-uniformity in light, aka a non-uniformity in radiative load across the leaf due to our leaf temperature measurement, given these different ways of measuring leaf temperature, which I think is kind of a cool error analysis that I hadn't seen previous to this.
No, agreed. It was super cool to see in the book.
Yeah.
That was a lot. That was 2 chapters shoved into hopefully what is around 30 minutes. It was a lot of surface level stuff. You're supposed to laugh. That was these are the jokes, people. That was a joke.
I'm going to make a plug since Seaton made a terrible joke about surface level. I'm going to make a plug for the last figure in the chapter on temperature. We took a little thermal imager and we shoved it into a chamber, and then we measured leaf temperature. Like we took an image of leaf temperature in a time series as we went from a dark acclimated leaf to a light acclimated leaf. And there's 3 panels from that little fun exercise in there. This is not a product available for sale. Don't take it as this, as Seaton squirms in his chair. No, this is just a fun exercise that nerds at LI-COR do when they have nerdy time to nerd out. And it's really cool because you get to see not only like, can you artifactually make gradients in leaf temperature, but there's real biological reasons why you get gradients in leaf temperature. And I think everybody should go find this chapter and look at this figure because it's so cool.
Agreed. And who collected that data?
Well, I was trying not to give Doug extra credit, but Doug absolutely did this.
Dougie Fresh did it. It's very cool. You should check it out.
I think when you get to the next chapter that Doug talks about, you need to ask him repeatedly who collected the data he's talking about in that chapter, just to make things feel balanced out after this this conversation.
I feel like I'm going to forget by then, so no promises. As always, we're going to end here with just a short 1 takeaway, and there's 2, okay, because I know what you're going to do. So in addition to reading the chapter as chapters.
Yeah, you should read both of them in order, probably.
I would at least read chapter 3 and then 7 and 8. What are a couple of takeaways?
I think one big takeaway is to make the discussion understandable. We do a lot of pretending in these chapters, particularly in chapter 7. Like, I don't know how many times we state this in there, but we pretend like things are isothermal. The world's never isothermal. Like, we rectify that when you get to chapter 8, but we do silly things to make it more understandable, we hope. Hopefully it doesn't just add extra confusion.
Nah.
And then the other takeaway, I think that, VPD. It's always VPD leaf. That's always the right answer. There's just not another answer. It's VPD leaf.
I love it. All right. Well, thank you very much for coming.
You're welcome.
Thank you, Jason, for joining me here today, and thank you, everyone, for joining us. Please check out Chapter 3, 7, and 8, as well as all the others. Because, again, this book is the only reason for this podcast series. So please do not forget to like and subscribe, read those chapters, and until then, stay curious, stay kind, and I'll talk to you soon.